I admit it. Catholics were right about Eucharistic miracles. Well, specific Catholics, anyway. I'm talking specifically about Doctors Stacy Trasancos and Dr. Kelly Kearse, to be exact. And they're right about Eucharistic miracles because they are Catholics who are saying things like important natural explanations were never addressed, and until those are ruled out as causes, it is premature to claim a miracle. They are also doing things like recreating Eucharistic miracles with unconsecrated hosts. And in 15% of unconsecrated hosts, they got a result nearly identical to real Eucharistic miracle results. In this video, I am going to dive into 2 new papers published in real journals talking about Eucharistic miracles. This video was born when a viewer sent me an article from Crisis Magazine, of all places. For those of you who don't know Crisis Magazine, I will quote from their About page. Crisis Magazine, About Us. Scrolling down a little bit. Crisis remains America's most trusted source for authentic Catholic perspectives on church and state, arts and culture, science and faith. We have one purpose and one only: to proclaim Christ's kingship over all things, at all times, to all nations. As you can imagine, I don't find myself agreeing with Crisis Magazine, like, all the time. But on December 23rd, 2024, just less than a month ago, Crisis Magazine published an article called Exaggerations and Eucharistic Miracles, written by Dr. Stacy Trasancos. This article from Crisis is primarily concerned with 2 new papers published in respected journals covering Eucharistic miracles. The primary author of both of these new papers is a certain Dr. Kelly Kearse. Dr. Trasancos makes a point at the beginning of this article saying that Dr. Kearse is not some kind of anti-Catholic radical or anything of the sort. He is a faithful Catholic, just one who cares about scientific rigor. Dr. Trasancos writes before summarizing Dr. Kelly's concerns, I want to make it clear that his purpose is not just to disprove miracles and not to question the real presence of Christ in the Eucharist. Quite the opposite. The present concerns address exaggerations and how to correct them. Kearse points out important natural explanations that were never addressed. Until those are ruled out as a cause, it is premature to claim a miracle. Dr. Kearse is a faithful Catholic, a Eucharistic minister for over 20 years, and a science teacher at Knoxville Catholic High School. But before that, he was an immunologist at Johns Hopkins, and he worked for the NIH, and he wrote a biology textbook. So the man seems like he's retired now, it seems, but he has his credentials. The first paper that the article from Crisis Magazine discusses is called The Relics of Jesus and Eucharistic Miracles, Scientific Analyses of Shared AB Blood Type by Dr. Kelly Kearse. This one was published in Forensic Science, Medicine, and Pathology on October 30th, 2024. Dr. Kearse writes, numerous artifacts have been described throughout history that were allegedly stained with the blood of Jesus, particularly those associated with his passion and death. He lists the Sudarium of Oviedo and the Shroud of Turin specifically in this article. I'm not really going to be discussing those. This is about Eucharistic miracles. He goes on to say, so the Sudarium and the Shroud both were reported to have blood type AB. Various Eucharistic miracles have been chronicled in the 21st century, in which consecrated communion wafers are described as exhibiting properties of human flesh and blood. For those tested, a similar AB typing was obtained, which has been suggested to imply a common origin among these materials as a type of reciprocal validation. So only a small portion of the human population expresses both A and B antigens, only 4 or 5%. Because blood type AB is rare, this has been used to bolster the claims that these Eucharistic miracles and the shroud and the sudarium all belong to the same person, because the odds of getting this 4 or 5% result time after time after time after time after time is about 1 in 3.2 million or higher. And that 1 in 3.2 million number, that comes from Catholic Answers. You can find this paper in the description down below and follow the footnotes if you would like. But how could it be coincidence that all of these objects exhibit the same rare blood type? Well, while A and B molecules were originally found on human red blood cells, it was later established that bacteria also express identical molecules on their cell surfaces. Because AB antigens are shared among humans and bacteria, one cannot be certain if typing results are authentic when dealing with aged or contaminated samples using these methods. A sample could test positive for AB without any real red blood cells even being present. Thus, when one considers that a common denominator among objects is the presence of bacteria, probability statistics as 1 in 3.2 million become much less impressive and scientifically shift to a different result. And so what that means is, imagine that, so you know statistically that the odds of all of them testing AB for human blood is 1.32, but you also know that bacteria can express AB. So what do you think is more likely? That these are all legitimately human blood, all AB human blood, or that they have bacteria on them? I mean, one of them seems way more likely than the other. Microbial contamination has been found in various instances involving modern Eucharistic miracles, as well as suggested to be the primary cause for many of these appearances. With aged and or contaminated samples, it is difficult to have confidence in the true nature of AB antigens that are defined using serological methods. In any of the above cases, the types of serological or antibody tests that were performed would not distinguish the true source of AB molecules, as they recognize identical structures in both bacteria and in humans. And then what's really interesting is that Dr. Kearse quotes from Dr. Serafini's book, the book that I've been reviewing this whole time. So Dr. Kearse writes, Dr. Serafini states that, quote, the overall risk of an incorrect blood group determination for these analyzed blood samples of these miraculous events is becoming increasingly small as methods have improved and have been carried out in various laboratories. This is an oversimplification of the fact that even though techniques may slightly vary, the molecular principles of antigen recognition by antibodies remains unchanged. As none of the above articles in question are sterile, quite the converse, it is reasonable to propose that the shared AB blood antigens from bacteria could readily explain the observed shared blood type. Scientifically, there are methods that allow one to distinguish whether AB antigens truly originate from human blood or from bacteria. Such techniques require examination at the DNA level, which has never been done for any of the aforementioned miracle events. Remember, we've been discussing DNA for these Eucharistic miracles, and every single time that we look at it, it's a miracle in itself that we can't replicate the DNA. Although bacteria and humans share common AB structures on the cell surface, bacterial GTA and GTB are distinct from their human counterparts. Thus, providing sufficient DNA was available, similar technology could be used to determine the true source, bacterial versus human, of AB antigens present in those substances. The findings that AB antigens have been found on various relics and Eucharistic miracles are by no means sufficient to support the claim of a single individual source. Demonstration of shared, unique, specific identifiers would also need to be present, and those unique identifiers are only present in DNA, and we've never found DNA on any of these Eucharistic miracles. How convenient. In summary, the most likely explanation for such observations is that they result from the presence of common antigens present on bacteria. Again, what is more likely? The 1 in 3.2 million chance that these are all legitimately human blood and they're all AB, or that they all have bacteria on them? The bacteria seems way more likely to me, and it seems way more likely to Dr. Kelly Kearse, a devout Catholic, and that's why he wrote this paper. And the second paper by Dr. Kelly Kearse, and this one with another Dr. Frank Ligaj, is titled Scientific Analyses of Eucharistic Miracles, Importance of a Standardization in Evaluation. This one was published in the Journal of Forensic Science and Research in November 2024. This paper is the one that includes the do-it-at-home kit for your own Eucharistic miracles. Dr. Kearse and Dr. Ligaj write, although not regularly verified, contamination with the bacteria Serratia maricenses, and I won't be repeating that because I'm sure that I butchered the pronunciation, has been suggested as a causative agent for the appearance of flesh and blood on communion bread. That's what I've been saying, and I have so many comments. Maybe I'll put some of them on the screen now that have been like making fun of me for saying this. But I don't think that those commenters realize how common contamination from fungus and stuff like that actually is when you're doing these kinds of scientific analyses on samples that were not collected in a hospital. Numerous other bacteria and fungi may also produce reddish products. Upon examine, and then Dr. Kearse and Ligaj go into each of the most popular Eucharistic miracles, and they discuss why they think contamination is likely. So they start with Lanciano, Italy. Upon examination in the 1970s, the sample was found to contain whitish specks, fungus, that showed conspicuous accumulations of spores and hypha of hyphomyces. And look, footnote 4 there is literally A Cardiologist Examines Jesus by Dr. Serafini. So Dr. Kearse has read Dr. Serafini's book and is quoting it to show that there was fungus. We identified fungus on the Lanciano host. In a recent book on the scientific nature of Eucharistic miracles, A Cardiologist Examines Jesus, Dr. Serafini states that, quote, it is qualitatively unquestionable that the miracle of Lanciano contains true human blood. Unfortunately, however, it cannot be declared that the results are specific for human blood, as very limited demonstration is given regarding the potential cross-reactivity of the anti-seria used, with only 2 other species tested, bovine and rabbit. Moving on to Buenos Aires. In this case, a host was found discarded near a candlestick holder towards the back of the church. The priest's first inclination was to consume it, but because it was so dirty, opted to place the wafer in water for future disposal according to church protocol. Several things should be noted. First, the primary experts that were sought out were pathologists or cardiologists, indicating that the assumption had likely been made that this was human cardiac. A more objective approach would have solicited additional input from scientists of other disciplines, including microbiologists and mycologists. Second, it would have been more valuable to conduct the study in a true blind fashion and mix in with the experimental slides various control slides prepared in the same manner. Although it is reported that Zugaba did not know the nature of the samples he examined, the presence of camera crew might have alerted him that these did not likely originate from an ordinary John Doe. That is exactly what I've said. And I actually have evidence that I don't think I've discussed on my channel yet, but Dr. Castañón Gomez submitted a sample, not for Buenos Aires, but for a different Eucharistic miracle, submitted a sample to a lab, and the patient's initials were IHS. Does IHS sound familiar? It should if you were a practicing Catholic at any point. Those are the first 3 letters in Jesus' name when spelt in Greek. And so my point is, I think that Dr. Castañón Gomez was keeping things blinded, wink, wink. I don't think he was actually blinding anything. I think that he was not saying it, but I think he was giving clear signs that this was miraculous in nature. Tests verified, and this is again Buenos Aires, tests verified the presence of human DNA, which was unable to be amplified, hinting at the divine nature of the sample. The report states that a very low concentration of human DNA was recovered, and that the sample contained high molecular weight, so good quality DNA, of non-human origin. The very low concentration of human DNA that was recovered is characteristics of contaminating DNA, the likely result of somebody touching the sample. It is also important to consider other possible non-miraculous sources of DNA that could exist in the sample, such as bacteria, fungi, and plants, like wheat. Indeed, wheat DNA has been shown to survive the baking process under relatively high temperatures and exposure conditions, and to be successfully amplified by PCR. Given the suggested involvement of microorganisms in the descriptions of miracles in the past, it would have been prudent to also consider DNA contribution from such microorganisms. But Dr. Castellan-Gomez did not. They move on to Tixla, Mexico, and they say that, so the nun did notice the red spot right away, but then they hid the host away for 3 years before any testing was done. That's a long time for fungus to grow. It is critical to emphasize that such tests are optimized for the use of liquid blood, which has a specific consistency and density and pH. Because certain Eucharistic miracles have been attributed to bacteria that produce a viscous, I don't know that word. It's probably mucilaginous or something, fluid. It is possible that the increased viscosity of the sample could result in nonspecific antibody binding, resulting in a false positive. As little to no details were provided for such tests, other than the result, it is unclear if differences in fluidity might be a factor. One pervading issue in many of these histological reports is that they are based on visual inspection alone. Histology, by the way, is when you look at it and you're like, yeah, that looks like heart tissue to me. So Dr. Kearse and Ligaj here are saying that visual inspection alone is a problem. While this may be suitable for normal routine of examining specimens from persons about which considerable information is known, identification of certain cell types or tissues in cases such as these requires accompanying studies with cell-specific markers. What he's saying there is, when a surgeon performs some kind of biopsy on a patient, they know what organ they are biopsying. And then when they look at it, they're like, yeah, that looks like the organ I just biopsied. Great, fantastic. Histologists aren't regularly, unless they work in like a forensic lab, they're not regularly getting samples that they don't know the history of. When these histologists who work in forensic labs get a sample and they legitimately don't know what it is, they don't rely on visuals alone. Yet that is precisely what we did in these Eucharistic miracle cases. And that seems to me to be a huge problem, especially because we only gave them to cardiologists. I mean, it's like shocking to me that anybody even takes these Eucharistic miracle investigations seriously based on that alone. To this end, the investigators reported that the samples were positive for glycophorin A, a red blood cell marker, but tested negative using probes for muscle desmin and myosin, which are both muscle specific. So they found a red blood cell, but they didn't find any muscle tissue. The presence of plant cells in the Tixla sample was acknowledged, recognized every now and then in the background. Again, quoting from Dr. Serafini's book here. Similar to Buenos Aires, DNA studies were done with the conclusion that the DNA was completely degraded and fragmented. Serafini imparts this once again is in keeping with the idea that the genetic material escapes genetic probes and does not lend itself to be recognized. The possibility of non-human DNA being present was not considered. Moving on to Sokolka, Poland in 2008. A host was noticed on the steps of the altar by a communicant, and because the wafer appeared dirty, the priest immersed it in a container of water. Later that same day, the contents were transferred to a larger container, and more water was added. A week later, it was noticed that the host had not dissolved and was covered by a reddish stain. The water was untainted by the color, an odd finding if dried blood or bleeding were involved. More on that below. But again, just notice here that it was found on the ground, transferred into water, and then a week later, the blood appeared. I'm going to skip Legnica, Poland, and Tyrol, Austria, because those aren't covered in Dr. Serafini's book. But taken together, these reports provide descriptions of shared features that have been ascribed to the common origin, most notably the suggestion that genuine blood seeped from consecrated wafers. In the specific analyses of Eucharistic miracles, it is interesting to speculate what might happen if a non-consecrated communion wafer were treated in a similar manner, as described for many of the above occurrences. Surprisingly, we were unable to find a single instance where such a basic experiment had been performed. Right, so that would be like the blinding stuff that he was talking about earlier. Never did Dr. Castellan-Gomez think to take an unconsecrated wafer, leave it out, and then include that in like as a blinding, and sending it to the various laboratories. Never. Here, we provide a novel demonstration that the appearance of a bleeding host can occur by placing non-consecrated wafers under similar conditions as described for many of these events. Using several fundamental scientific techniques, major differences between fresh blood and ensuing reddish areas were denoted. Multiple sources of non-human DNA were shown to be present in unconsecrated wafers, providing an explanation for previously failed amplification attempts with miracle wafers. Finally, a minimum protocol is suggested to aid in the standardization of evaluation and scientific validation of such cases in the future. Okay, so this is what they did. This is the make your own Eucharistic miracle at home kit that the rest of this paper is devoted to. So, wheat communion wafers were purchased from Kavanaugh Altar Breads Company in Greenville, Rhode Island, a common supplier for many parishes in the United States. Wafers were left in a dusty and dark corner for several days. Samples were then placed in approximately 200 milliliters of tap water in plastic 16-ounce solo cups and cultured for 7 to 10 days at ambient temperatures and humidity. Great. So that's the control for the host. Now, these are unconsecrated. So theoretically, a miracle should be impossible. Well, maybe not impossible, but why would God choose to perform his own transubstantiation, I guess? Why would he do that to an unconsecrated wafer? That would seem strange. So now the researchers wanted to collect fresh blood, real fresh blood, to compare. So fresh blood was obtained from healthy volunteers. Then they also wanted to get some known fungus so that they knew what they were looking at. So they bought purified, and then here's Serratia M, that culture that they discussed at the beginning, and that was purchased from Carolina Biological Supply Company. They took some of so plate cultures were swabbed with sterile inoculation loops and transferred to wheat wafers placed on damp paper towels and incubated at room temperature and humidity. So there's a bunch of different things going on first. So they bought a whole bunch of unconsecrated wafers. They took a bunch of them and left them in dusty corners of random rooms, and then put them in water after 24 to 48 hours, and then left them for a week. For a different amount of them, they actually just straight up inoculated them with the fungus, and we can see, will they compare, will they look similar, the ones that we left out and the ones that we purposefully inoculated. And then they also grabbed fresh blood from human volunteers. Interestingly, reddish growth on the wafers was apparent within 24 to 48 hour time period of the ones that they inoculated. So in each of the ones that they inoculated with the fungus, they found reddish fungal growth. Then they decided, let's do some DNA extraction on the plain old uninoculated hosts, and they found wheat DNA. That is what it is saying here. They found wheat primers, and they're actually listing the DNA sequences right here. But that just goes to show you that even without the fungus being present, you can find DNA that won't be replicated, and you can find human DNA, right? If it's just been handled by somebody, you'll find a small amount of human DNA and then a lot of non-human DNA. And so on completely unconsecrated, no blood was placed on it, no fungus was placed on it, you can find exactly the same results that you found in these miracle cases. I just thought that was really interesting. Also, right here, I just wanted to highlight Roche, Thermo Fisher, and Eurofins. These are real labs, and one of my concerns with the work that was done by Dr. Castellan Gomez was picking out individual scientists who work at laboratories and universities and asking them to do the work. Oh, and by the way, these people also, like in the case of Dr. Zugaba, happen to be published Catholic authors. Oh, what a funny coincidence. Why not just send them to Roche or Thermo Fisher or Eurofins, where you won't know the name of the scientist performing the test because the name of the scientist shouldn't matter. But that never happened in these Eucharistic miracle cases that we looked at. The scientists were always hand-selected to be Catholic-friendly scientists. Okay, so here are the results. Going back to the non-consecrated wheat communion wafers that were placed in various areas on or near the floor for several days and then transferred into containers of tap water, these samples were kept indoors, undisturbed at ambient temperature and humidity, and away from direct sunlight. In approximately 15% of these cases, a bright red area was noticed growing on the remaining wafer portion some 7 to 10 days later. A 15% success rate, just leaving unconsecrated hosts around and then leaving them on the ground for a couple of days, leaving them in water for 7 to 10 days. 15% success rate. I mean, that's insane. That alone should, frankly, be the death knell for claims of Eucharistic miracles. In the study of Eucharistic miracles, it has been reported that the water remained untainted 1 week later for the host after the host was immersed and the red stain became apparent. This is intriguing, as fresh blood or freshly dried blood is readily dissolved in water and many aqueous-based solvents. In the current study using non-consecrated wafers containing reddish areas, it was noted that the water remained untainted as well. Freshly dried blood is readily soluble in aqueous solvents, but the reddish area on the experimental wafers remained insoluble. Okay, if you don't know what those words mean, if you take blood and you put it into water, and then you come back 24 hours later, the blood won't be in the same spot. It'll have dispersed throughout the water, right? So now, depending on how much blood you put in there, you'll have like a uniform, homogeneous, pinkish liquid, which is 99% water and 1% blood or whatever. If you only put a tiny drop of blood into a large thing of water, you won't be able to find the blood droplet anymore, and the water will still be clear. That is what they're referring to with that word soluble. Blood is water soluble. In all of these Eucharistic miracle cases, the blood did not dissolve into the water. Rather, it kind of all clumped together in this weird viscous liquid, and it sat on top of the water. These unconsecrated wafers did the same thing. The reddish stuff clumped on top of the water and did not disperse itself into the water. It was not soluble like you would expect it to be. As that bacteria that we've been discussing has been frequently suggested as a potential causative agent in Eucharistic miracle events, wafers were inoculated with a purified culture of this bacterial stuff, and then within 24 to 48 hours, they found that on there. So on 100% of the ones where they purposefully inoculated it, they got the results they were wanting. And 15% of the ones where they just left it at, they got the same result, which is super interesting. Scrolling down, we can actually see some of the pictures of the work that they did. I'm not going to get into the ultraviolet fluorescence part of it, but ultraviolet fluorescence is something that could be done to determine whether or not, to rule out certain bacterial substances. And to the best of my knowledge, this kind of ultraviolet fluorescence work was not done on any of the Eucharistic miracles. Taken together, these data indicate that the reddish areas on non-consecrated wafers resulted from fungal growth. In addition, these data show that a general examination under ultraviolet light can prove useful in the initial characterization of enigmatic reddish areas and may also provide further diagnostic information for various past events. In previous miracle cases, the non-amplification of DNA in human-specific primers was suggested to represent a type of divine property. We reasoned that 3 potential sources of non-human DNA might exist under such conditions: DNA from the wheat wafer itself, DNA from bacteria, and DNA from fungi. Whole DNA was very fragmented in the unconsecrated wafers, but was sufficiently intact to be amplified using PCR, giving DNA product of expected size. The sequence aligned with wheat bread. Unsurprisingly, right? We found wheat DNA in an unconsecrated communion wafer. But the point is that they found non-human DNA that couldn't be amplified. It's probably wheat DNA in the Eucharistic miracle cases too. Whole DNA was also purified from the cultured wafers with the reddish growths, which showed a much stronger signal compared to the non-treated wafers. So that could be the high-quality non-human DNA that we saw in a lot of these Eucharistic miracle cases. Fungal primer samples produced a consensus sequence that was 313 base pairs long, which matched several fungal sequences in the gene bank with a 99.35% identity. So they know that what they were looking at was fungus. Definitive sequencing to identify the specific species of fungus present in the cultured wafers, though, was beyond the scope of the study. Taken together, these data indicate that multiple sources of non-human DNA could be isolated from the cultured wafers, including those from plants, bacteria, and fungi. Importantly, these results identified this certain family of fungus as the most likely cause for the observed reddish area growths in our study. While much attention has been placed on the evaluation of consecrated hosts, suggested to exhibit miraculous properties of visible blood, we show here for the first time that ordinary, non-consecrated wafers show similar appearances when treated in like manner. Similar to what has been described above for various miracle hosts, we found that the water in which the wafers were placed did not become tainted over time. If real blood were being issued forth, that would be a relatively odd property, as blood should be readily solubilized. We found that only about 15% of non-consecrated wafers cultured in water developed the reddish blood-like areas, supporting the idea that reports of miracles are relatively infrequent and do not occur every time abandoned hosts are disposed of according to church protocol. Although our studies did not implicate this specific fungus as a causative agent, it is understandable how common this infectious agent of bred substances could produce the appearance of a bleeding host. Molecular biology studies of miracle wafers have been attempted, particularly short tandem repeat analysis, which has failed to produce any results and thereby attributed to an enigmatic and divine nature of the DNA involved. As shown in the current report, the prevalence of DNA from other species, plant, bacterial, and fungal, in non-contaminated wafers offers a plausible alternative to the findings of previous Eucharistic miracle investigations. Evaluation of HLA markers also provides a vital cross-check for the submission of fraudulent samples. An identical profile should be obtained in all instances worldwide if the claim of a single origin is legitimate. This is very important. And look, they italicize that to stress how important that is. And these HLA markers have never been found in any, not the shroud, and none of these Eucharistic miracles, not the sudarium, none of that. This is very important if the scientific community is to take this stuff seriously. Finally, the authors wish to state that the purpose of this paper is not to suggest that Eucharistic miracles should all be viewed as fallacious. Rather, the aim is to emphasize that solid scientific practice should be used in the evaluation of such occurrences so that a true assessment may take place. Faith and science need not contradict one another. However, one of the major issues with the subject of Eucharistic miracles is that no standard scientific protocol exists for their examination. To this end, a suggested minimum procedure is described below to augment any future investigation and serve as a starting point to standardize scientific evaluation of such occurrences. It is also foreseen that additional criteria may be included to aid in the improved reproducibility and reliability of the results. The following section is, they're just suggesting different tests that you could do to make sure that what you're looking at truly is from humans and not from fungus and not from wheat. I wanted to highlight this, that the normal course of action in any scientific investigation is to write up the results for submission to a scientific journal so that the findings may be critically and constructively evaluated. Scientific transparency is important for the establishment of the belief that such extraordinary events might be possible. Premature reporting by press release of incomplete conclusions should be avoided. Relatedly, liturgical representatives should be particularly diligent in fact-checking the scientific claims that often around such events and update any current websites and publications regularly. In summary, the current report has evaluated the results from various Eucharistic miracles with particular attention to the methodology used in the analysis. Additionally, evidence was provided that the appearance of a bleeding host can occur by placing a non-consecrated wafer under similar conditions as described for many of these events. Distinctions between intuit red areas and genuine blood on experimental wafers were noted, and ultraviolet light was shown to be a useful discriminator. Our studies indicated that the presence of particular fungus being responsible for the reddish growth areas on the wafers suggests towards establishing a minimum protocol of scientific examination were put forward to help standardize the investigation of possible miracle occurrences in the future. And that's the end of both of the papers. I would be really curious to get Dr. Linole's opinion about these two papers, especially since Dr. Kelly Kearse cites Dr. Serafini's book and disagrees with Dr. Serafini's book. And they're both Catholics. I don't know how to get in contact with Dr. Kelly Kearse, but I might try if I can figure it out. And I don't know, maybe... I know that my friend Dale from Real Seekers knows Dr. Serafini. Maybe we could put together a conversation if, I don't actually have a way to contact either of these people. So, this is a little bit pie in the sky dreaming. But I think that would be really cool if I could. It would be awesome to get two Catholic scientists, right? One researcher and one doctor. But getting them on the same call to discuss these Eucharistic miracles on which they disagree, that would be an awesome conversation. But until that time, thanks so much for watching. And I'll catch you in the next one.